Well, I too would like to thank the organizers. I'm up here really as a representative of a group that Linda Bonewald put together, myself, Linda, and Marco Brado, a team of both bone and muscle biologists. And so my work for several years now has been on the role of Wnt signaling in bone. And the outline for my talk today is just to give you a little bit of background on the importance of the Wnt beta-catenin pathway. I think this is the first meeting I've been at in a long time where no one else has talked about Wnt before me, so it's kind of novel. And then I'm going to talk about work that we've been doing recently to really try and dissect out the importance of the Wnt beta-catenin signaling pathway in mechanical loading, because it's in the context of mechanical loading that I'm going to be talking about the muscle-bone crosstalk mechanism. I just want to mention briefly, and this has already been done eloquently by many of the other speakers about muscle and bone as endocrine organisms. There's some other issues that really haven't been discussed that I want to talk a little bit about. And then I'll talk about muscle-to-bone crosstalk, and finally bone-to-muscle crosstalk. So this is the Wnt pathway. It's a rather complicated pathway. This is a very simplistic diagram. It has over 200 players involved that we know of right now. And the basic point of the pathway is to control this molecule, beta-catenin, and its intracellular levels. Normally, beta-catenin is kept at very low levels, and it's degraded through a series of phosphorylation events that's regulated by this degradation complex, and where it gets phosphorylated, ubiquitinated, and then degraded in the 26S proteasome. But in the presence of Wnt ligand binding to the co-receptors LRP5 or LRP6 and the frizzled family of proteins, a series of associations and phosphorylations occur, which ultimately leads to the inhibition of GSK3 beta, the dissociation of this complex, and the release of beta-catenin into the cytoplasm. And we now have some evidence to suggest that beta-catenin is escorted into the nucleus by estrogen receptor alpha, probably in a ligand-independent mechanism. Once inside the nucleus, there's any number of transcription factors, principally the TCF-LEF and FOXO family, that compete for the binding of beta-catenin to control gene expression. So what do we know about the importance of Wnt beta-catenin signaling in the skeleton? Well, there's actually been quite a lot of work done over the last decade or so in this regard. We know that it controls osteoblast versus chondrocyte differentiation during skeletogenesis, favoring osteoblast differentiation. It controls the balance between osteoblastogenesis and adipogenesis, and it's also important in osteoblast proliferation, differentiation, bone matrix formation, mineralization, and apoptosis in bone cells. It also plays an important role in osteoblast control of osteoclastogenesis through the regulation of rank ligand and OPG. And there's been some really beautiful work recently on its role in fracture healing. And a number of groups, my group and many others, have looked at the role of beta-catenin and the Wnt signaling pathway in the response of bone to mechanical loading. So I'm going to talk a little bit about the osteocyte mechanical loading, but I would like to echo the comments of Clint Rubin earlier. Every cell in the body will respond to some kind of mechanical stimulus. But I would also add that there's different sensitivities, and the different forms of mechanical stimulus may be different from one cell type to the next. And within bone, the osteocyte makes up over 90% of the bone cells. They're viable for decades, and they are buried deep in the mineralized matrix. This is a resin cast acid etch, SEM. So what you're seeing here is actually the lacunar form where the osteocyte body dwells within, and then the canaliculi where the dendritic processes from the osteocyte extend outward. And these dendritic processes form gap junctions with adjacent osteocytes, and they also make interconnections with cells on the bone surfaces. And it's because of its location in bone and its relative abundance and what have you, that for many years people have postulated that it's the ideal cell in bone to sense mechanical load and translate strain or lack of strain into a biochemical signal that then alters the behavior of cells on the bone surfaces. Osteocytes produce several factors which are key regulators of this Wnt pathway. You heard about one a little earlier today called sclerostin. Osteocytes also produce DKK1 and the secreted frizzled-related proteins. All of these inhibitors act in some kind of an equilibrium, dynamic equilibrium that we understand, to block the action of Wnt. Remember, the osteocyte does not divide. So one of the normal functions of the Wnt pathway in intestinal cells or whatever is proliferation. If the osteocyte were to divide, it would die because it has a physical space constraint. So this pathway is kept off as much as possible through the action of these regulators, and they all act in different mechanisms. I mentioned sclerostin just briefly. It's been an interesting gene to study. Deletion or mutations in sclerostin result in human conditions known as sclerostiosis or Van Buchen's disease. Mice that lack sclerostin have increased bone density, bone formation. It's a potent inhibitor of osteoblastic bone formation, and it has autocrine effects in osteocytes to stimulate rank ligand expression, which is work that's really been very provocative that's come up just recently. It's also been shown to be expressed in articular chondrocytes, and several people have looked at the regulation of sclerostin. Our group, Alex Roebling, Terry Bolito, and others have looked at its regulation by anabolic loading and PTH and various other things. This is just immunostaining. You can see the brown staining here corresponding to the presence of sclerostin. And all of these cells that you see here within the cortical bone are the osteocytes. Interestingly, in our hands and many other labs, not every osteocyte is always sclerostin positive. There seems to be some heterogeneity in the level of sclerostin production by different osteocytes. So it may be actually a marker of a more mature or later, longer-lived osteocyte rather than a newly embedded cell. Okay, so how did we study the role of the beta-catenin signaling pathway in the osteocyte? Well, we're using a reporter mouse called the top-gal mouse. This is a mouse that has the LAC-Z gene, which expresses beta-galactosidase, under the control of a promoter that's driven by beta-catenin. So when beta-catenin translocates into the nucleus, this mouse will express beta-galactosidase, and you can stain for that, and you get a nice blue-colored cell. So we take the right forearm of our mouse, and we load it for a very brief 50-second loading session, and we use the left forearm as an unloaded control. We have a group of mice, and we sacrifice 4 of them 1 hour after this 50-second load, 4 hours, 24 hours, et cetera, et cetera, et cetera. And then we isolate the bones, fix them, stain them, and then we section them from the tip of the olecranon all the way to the wrist. You get about 1,000 sections or so out of these mice for each of this, and we can map the entire lacunar network in terms of which cells have activated beta-catenin signaling as a function of time. And so when we look at the kinetics of this, what we see is that rapidly, 1 hour after, as early as 1 hour after that single loading session, I've lost my pointer, a few osteocytes have turned blue, expressing the gene. By 4 hours, we're starting to see more, and by 24 hours, we're seeing even more. We can take these same sections and we can stain with antibody against sclerostin or DKK1. This just shows a merge. And then we can analyze the number of cells that have expressed beta-catenin or what the changes in sclerostin and DKK1 are. And what we see is a very rapid increase at 1 hour. reaching a peak by 24 hours and then a return to baseline by 72 hours in terms of the expression of beta-catenin, or of the reporter gene, I should say. At the same time, maximal changes in sclerostin and DKK1 are observed at 24 hours. So the kinetics of activation is much faster than the loss of these inhibitors appears to be. And so basically, just to summarize, after 1 hour of loading, beta-catenin activation is observed. It reaches a peak at 24 hours. The sclerostin and DKK decreases are maximal at 24 hours, also at a time when beta-catenin is maximal. And they return to baseline at 24 hours. And I didn't mention or show you the data, but we don't see osteoblasts on the lining surfaces activating the pathway until at least 24 hours, which suggests that the osteocytes, the early responder to mechanical load in this system. But then that raises the question, so is all this pathway activation really leading to anything? So we asked the question, is beta-catenin essential for the anabolic response of bone to load? And the way we did this was to turn to a genetic model in which we took beta-catenin floxed mice and crossed them with the DMP1 Cree mouse. And the DMP1 Cree mouse is targeted mainly to osteocytes. And this was work that had been previously done by Dr. Bonewald and Michaela Kniesel's group at Novartis, had demonstrated that when you do a homozygous deletion and you completely delete, by 8 weeks of age, these bones acquire this really moth-eaten appearance. And in fact, if you were to try and load these bones, you would just break them because they're so fragile. So we had to choose to use for these loading studies a heterozygous deletion in which we deleted one allele of beta-catenin. And those mice in the work by Kramer and Bonewald and Kniesel appear to be pretty normal. There's a slight reduction in BMD, but you don't see any of this aberrant osteolytic-like lesions in those animals. And so this is just showing you the female data. But here's a double labeling experiment in which we loaded animals over the course of 3 weeks, did a calcine and alizarin red injection. And you can see in the control animal, the left limb is a very quiet surface. This is a 16 to 19-week-old animal. The right forearm builds a very robust bone formation response. And in the heterozygous knockout, there's very little bone formation taking place. And this just quantitates it by mineral apposition rate. Again, you see a nice robust bone formation response either on the endocortical or periosteal surface in the control, but in the knockout, very little. So what that tells us is that beta-catenin is essential for the bone anabolic response to load. And while one allele appears to be necessary for normal bone development, it takes both alleles to be are required for an anabolic response to load. This just summarizes that work. I haven't shown you all of this, but I hope I can convince you that the osteocyte's a mechanical responsive cell. It's involved in osteocyte, and the pathways involved in osteocyte communication, viability, integrity of bone, and bone formation in response to load. And through those studies and a lot of other studies, we put together a model to try and explain the difference in kinetics between the disappearance of sclerostin, the activation of the pathway, and other things. And what we're suggesting is that when you load bone, you have fluid flow through the lacunar canalicular system. You get the release of PGE2, which binds to its cognate receptor, activates AKT, which can inhibit GSK3 beta. You get beta-catenin into the nucleus. That results in decreases in sclerostin and DKK1, increases in Wnt, and you get a sort of a feedback then at the level of the receptor. This has been discussed and published in a couple of papers. But this then gives us the targets that I'm going to talk to you about in the remaining time, the assays that we use to look at bone muscle crosstalk. And that involves beta-catenin nuclear translocation, AKT activation through increased phosphorylation, and PGE2 release. So the traditional view of muscle and bone is that muscle loads bone, and bone provides an attachment site for skeletal muscle. And we know from all the talks at this meeting that osteoporosis and sarcopenia are major clinical problems. And as Tammy Harris elegantly pointed out, muscle atrophy alone can't fully explain the totality of osteoporosis, and reciprocally, aging-associated decreases in bone mass don't fully explain sarcopenia. And so what we have hypothesized is that there's an endocrine loop, that muscle produces factors that somehow sensitize bone, in particular the osteocyte, to mechanical load, and that the osteocyte produces factors that somehow alter muscle function, and probably other cells like the osteoblast. So is there any precedence for this model? Well, the osteocyte is an endocrine cell, and you've heard a lot about the muscle myokines, but the osteocyte produces FGF23, MEP, FEX, DMP1, and sclerostin that have the ability to either act locally in bone or on other target tissues. But how can these factors from bone and muscle reach the other tissue? That's a big question that I don't think anyone has really addressed at this point. Remember, bone and muscle are intimately associated tissues, and so it's possible that there could be just direct perfusion or diffusion of molecules between the two tissues. But by the same token, the osteocyte lacunar canalicular system is intimately associated with the vascular network, and some of these dendritic processes actually extend into these vascular spaces. And we know from studies using Procene Red that if you inject this into the tail vein, within 10 to 15 minutes, the entire lacunar canalicular system is filled with that dye. And in fact, molecules as large as 70,000 kilodaltons, such as BSA, can travel through the lacunar canalicular system. So let's look at the muscle to bone crosstalk. And our hypothesis here is that muscles producing factors that will affect bone function, and that these factors may interact or synergize with mechanical loading. So to test this, we've used the differentiating C2-C12 myoblast to myotube system. These are the myoblasts, these are the myotubes, and we take conditioned media from each of these sources and then apply them to the MLOY4 cells and primary bone osteocytes to see the response. And this just shows beta-catenin nuclear translocation. When we add 10% conditioned media from myotubes, we get very nice, robust beta-catenin nuclear translocation. It titrates down, but interestingly, myoblast conditioned media has no effect. So this is a factor that's being produced by the myotubes. This is under static conditions, understand. We've looked at AKT signaling upstream, and we see that as these myotubes differentiate over time, the factor increases in concentrations, leading to increased AKT activation through phosphorylation. And this actually is under static conditions is a transient effect. It peaks at around 30 minutes and by 2 hours returns to baseline. And this was important to determine because we needed a time when we weren't superimposing the effects of fluid flow on top of these transient effects. So we wait 2 hours after the addition of media before we do our fluid flow studies. And when we do that, we see actually that myoblast conditioned media has no effect on the fluid flow activation of PGE2 release, but myotube conditioned media actually potentiates the release of PGE2. At the same time, myotube conditioned media also enhances the phosphorylation of AKT above what fluid flow does by itself. So there's a factor coming from these myotubes that's potentiating the effects of fluid flow shear stress on these osteocytes. We've also looked at Soleus and EDL muscle fiber types, and interestingly, the Soleus conditioned media, when it's been stimulated in culture, has no effect on activation of AKT signaling in the Y4 cells, but the factor coming from the EDL actually potentiates the effect of signaling. So what I've just shown you is changes in PGE2 production and signaling, downstream signaling consequences as a result of the presence of myotube. And that myotubes are producing a factor that enhances the load response, and so does the EDL muscle. So Katerina Jan at this meeting has shown some very nice data showing that the myotube conditioned media also protects against dexamethasone cell death. I'm just going to refresh your memory on that data. This is dexamethasone-induced cell death above control. When she adds myotube conditioned media, she can block that effect. And when we knock down beta-catenin signaling, we actually lose that effect. So it appears that the conditioned media that she is now pursuing the identification of that factor with is actually working through this pathway. Okay, so let me just wrap up then quickly with some bone-to-muscle crosstalk studies. And again, here we're looking at osteocytes producing a factor that affects muscle function. And this is all review. You've seen this before. Conditioned media from osteocytes, when we isolate it from young animals, these are 4-month-old, actually increases contractile force in these myotube fibers. But when we use aged conditioned media, we see no effect. So what's the responsible party for this? Well, we're looking at PGE2 and Wnts, obviously. And when we've looked at conditioned media from MLOY4 cells subjected to fluid flow, we get a nice increase in the differentiation of these myotubes, and PGE2 will mimic this effect, whether it's in myotube number or size. We also see that conditioned media from MLOY4 cells induce calcium transients in these cells, and that this effect can be mimicked by PGE2 and blocked by an EP2 receptor antagonist. And that conditioned media from the osteocytes also induces beta-catenin nuclear translocation as part of this differentiation process, and you see that going on here, reaching a peak at 60 minutes. We've also done primary osteocytes, not just the cell lines, and when we isolate primary osteocytes from young animals versus aged animals, we see a very nice effect of beta-catenin nuclear translocation in the young and a blunting, almost an inhibition from aged animal osteocytes subjected to fluid flow. And so we've also tested the effects of Wnt3A in this model system. And we see again these calcium transients being induced. We also see a peak increase in contractile force over a period of time in response to Wnt3A. And when we've done gene array studies and we've looked at the effects of conditioned media, PG2, and Wnt3A, we see that these three things all produce very similar results in the myogenic program, inducing genes that are involved in myogenic differentiation and the contractile machinery. And just to show you one piece of in vivo data, we looked at the muscles from our high bone mass transgenic mice. This is an animal, a mouse model that carries the G171V mutation in LRP5. It's a transgene that's under the control of the collagen promoter, so we don't see any expression of it in muscle. And yet these animals have much higher contractile force in their muscles due to a gene that's being expressed in bone. So we have some evidence, I think, now from our conditioned media studies to suggest that PGE2 and Wnt3A and the Wnt pathway are also involved in crosstalk between bone and muscle. So that leaves us with some questions. What are the factors? How do they exert their effects? And more provocatively, can we rescue function in the aged animals where we're seeing these deficits by simply replacing one half of the pathway, or do we need to replace both halves of the pathway? And there's a lot more, obviously.